Compositional design is the topic of what we make our devices out of. It's the process by which you choose the parts that will make up your system. Because such tools must make choices about which parts to use or not use, such tools must operate with a currency of information about parts. There are 5 different types of information that are usually encoded by a software tool in this category. Grammatical information says something about the part's role in the central dogma. For example, this promoter part initiates transcription. Biochemical information says something about the molecular details of the interactions encoded by the part. For example, the promoter is bound by Ara C. In some tools, more granular knowledge of the energetics and thus kinetics of the biochemical interactions must be captured. For example, the Kd for Ara C bound to the promoter is 50 nanomolar. Other systems allow users to define families of related parts, such as useful sets of promoters or mutants of a gene. For example, this part is a member of the inducible promoter family. Finally, some tools allow you to define phenomenological or experiential rules about how a part should be composed with other parts. For example, don't use PBAD to drive part E0043 because it will kill the cell. The organization of information in the context of computer science is referred to as an ontology. Thus, there are part ontologies that form the basis for many tools based upon these different ideas of what is the relevant piece of information for computation. We've already talked about GenoCAD briefly. It uses grammatical types like promoters, coding sequences, or terminators to organize its parts. When you design a construct in GenoCAD, you first design at the level of abstract compositions in which each position of your part could be any of a list of promoter or any of a list of terminator or the like. You then can concretize each position of the device by using a pull-down menu and selecting the ones you want. Eugene was developed by Douglas Densmore, and it encodes user-defined families of parts and user-defined phenomenological rules of composition. In the example here, we illustrate 2 abstract compositions made of families of parts such as a passenger or a PP, which is a pre-pro sequence. These user-defined families are each a set of concrete part sequences. You encode a Eugene script by defining devices in terms of the families which make up the parts that have properties that are expressed in primitive data types. In this way, you hierarchically describe the space of a composition and the properties of the potential constituents of that space. The script also encodes rules about which parts can be combined with other parts. For example, it is experientially known that when you encode a P450 oxidase in the cell, you need to provide a reductase from the same organism or they will not interact. You can express the source organism as a property when describing the device, and you include this rule of source organism as shown here. Once you have written a script, you can execute it. It will permute all members of the abstract device and output a list of concrete compositions that all satisfy the supplied rules. Tools that attempt to design metabolic pathways from biochemical ontologies have been under development for many years. Here we show the sort of output these tools can generate. This is all the paths that Axe Synthesizer finds for making pyraminophenol. Axe Synthesizer was developed by a Berkeley team, including myself, which we have used in this course. Axe is a more recent upstart in this category. Other major efforts in this area are Vasily Hazanmanikadis' program BNice and PathFred. All are based on tracing paths through data encoding enzymatic information, such as butanol is oxidized to butanal by Felzy oxidoreductase, either directly or indirectly. The most commonly used tricks are a breadth-first search algorithm to trace paths to a target through the hypergraph of reactions, or to employ reaction operators that capture the pattern of atoms present in these reactions. It turns out to be quite meaty and complicated task to design biosynthetic pathways, hence the many efforts to solve it. It is challenging both in terms of how we represent biochemical information and the computational challenge of searching through a very large hypergraph. All the tools I've shown here fit in the general realm of compositional design. They deal with the question of how you go from a specification of behavior to a specific part composition with a specific DNA sequence. The tools I've described each have a different niche within the design flow and will likely ultimately be combined into a singular coherent toolchain. The first attempt at unification was the TASB effort, in which a high-level specification language called Proto, developed by Jake Beal, could be reduced to an abstract genetic regulatory network, which in turn could be concretized with distinct parts, then assembled in combinations by automated assembly. In this context, the retrobiosynthesis tools are comparable to the high-level role of Proto, but for a different use case and a very different mechanism. It has become clear that at this very high level of design, different synthetic biology devices, the biosynthetic devices, cell surface display, biosynthesis, etc., the software challenges of synthetic biology are best solved in domain-specific ways. However, below this level, at the part specification level, the software all likely collapses into a singular workflow.